Okay, thank you very much. Thanks all for coming. My talk today is going to focus on a project that is called PerMedCoE. PerMedCoE is a center of excellence. So the center of excellences are like a kind of special projects, European projects, that try to bring HPC, high performance computing, to different fields in science. In our particular case, of course, is personalized medicine. So today I'm gonna describe or summarize all the activities that we were doing during the almost 3 years of project. We are trying to do like a second iteration that will explain also like the main goals that we have in this project, and I hope you enjoyed this presentation. Okay, in the PerMedCoE we have like different institutions in Europe. Some of them are more focusing on the biological part in personalized medicine. Some others are more focused in the computational part. And this mix of different fields what makes really a strong this kind of initiative, because we can understand like the different limitations and benefits from all the points of view. So our context, of course, is the context of personalized medicine. We are approaching the personalized medicine from a very particular point of view. In this case, we are like working in this almost classical paradigm that tries to describe that, for instance, you have a patient that arrives to a hospital, you take a sample, and with this sample, you build a model, you train a model that you can use in order to ask like many questions in silico, right? You can like do like a high throughput, for instance, a screening of different drugs. You can try different combinations. And then once you select the best option, the best treatment, you apply this. So this is like a future solution that I hope we will have all together. And how we approach this problem. We usually work in simulations. We try to simulate tissues. We try to simulate tumors, for instance, growing, in order to have like this kind of information, because there are many diseases where the tissue structure or the tissue distribution is like really important. So the tool that we usually employ to do this kind of cell level simulation is PhysiCell. And one of the main tasks that we are doing here in this project is to improve this tool, to extend this tool in order to improve the scalability in professional HPC environments. With this tool, we not only simulate how the cells interact, we also simulate like different molecules that are diffusing across the extracellular space. And this could be like simple molecules, like for instance, oxygen, but we can also simulate like different drugs that are targeting, for instance, tumor cells. In this case, you can simulate how, for instance, a tumor is growing, or how a given drug could affect a specific cell type. So we are also extending this approach in order to introduce molecular pathways into every single cell. The idea is to base the behavior of the cells in the virtual population based on what is already now in the databases, what is already now in the literature. So we usually employ this molecular pathway that describe how the proteins interact, or how the molecules, the different ligands that we have at the extracellular space interact to define the main outcomes in the cell, like for instance, cell death or cell proliferation. And this is like one step beyond of the classic bioinformatics, right? Usually we use molecular pathways to infer like many different effects in diseases, in order to infer also, for instance, the effect of a specific drug in a specific target. And what we are doing here is to extend this, to embed this into the simulations, to take into account how the tissue is distributed, or how the different resources that you can have, for instance, in a tumor, are playing an important role in a disease. So this is a very useful approach that you can use to simulate, for instance, drug regimens, to simulate like systems that are quite dynamic. If you want to do like simulations with millions of cells or even billions of cells, recapitulating the very complex features of real tissues, and you want to introduce this molecular pathway, you want to have like different cell types, of course, at the end of the day, you need high performance computing, right? You need a lot of computational resources. And we consider that this is the kind of future in personalized medicine that we will need to reach this kind of digital twins that is like the definition or the term that we are usually employing in this field. And for doing this, we have selected different tools that are widely used in the community, apart from PhysicCell that I was mentioning. We have Mabos, we have Carnival, and we have Cobrexa. They are very useful tools to define like this molecular pathway that then we use in the simulation. For instance, Mabos is really useful to represent molecular pathways as Boolean models. We also have Carnival to infer these signaling pathways from transcriptomics, for instance, and we also have Cobrexa for working with genome scale metabolic models. And it is a nice tool to, for instance, do a high throughput screening of mutants. So we're employing like different strategies to improve these tools. The idea is to let these tools not only to do parallel computing, but also to do distributed computing, meaning that for a given simulation or for a given analysis, you can use simultaneously different machines. So this opens the door to have like models or to have like simulations or digital twins that are quite efficient. This is what we are pursuing in the future. And it is a matter of what is the strategy to use, but also it's a matter of detecting bottlenecks in the source code, like parts of the code that are working very well when you have like a low scale problem, but when you try to expand this to using like 100 machines, immediately you find like different things that you didn't realize, right? So this is a task that was done also in collaboration with the experts of another center of excellence that is called PopCoE that works in detecting this kind of bottlenecks in the code. We are, as I was mentioning before, we are using the different strategies. The tools that we are selecting are quite different. Even they are using different programming languages. We have Julia, we have R, we have C++, for instance, and depending on the solution, depending on the tool and the use that we made, we have to select a different strategy. We were using, for instance, MPI for Mabos or for PhysicCell, but we are also using Julia. We are also refactoring some parts of our codes in order to be compatible. We are also using GPUs in order to optimize some parts of our codes. That it will be probably an important part of the future in PerMedCoE. And we are also connecting these tools. These tools are like building blocks that are really useful to solve real use cases. You, for instance, think using PhysicCell to simulate a particular use case or a particular disease, for instance, probably you will need some omics data. To personalize the models, probably you will need like some preliminary analysis, and then you will do the simulations. And probably if you are applying the same for 100 patients, you will need to do like a kind of meta-analysis to understand what are the differences, for instance, between two groups of patients. So when you have to do this, immediately you start thinking of workflows. Of course, it is like the standard computational solution that we have. In this case, we are using standard workflow managers, like for instance, PyComs. PyComs is a workflow manager that is also being developed at the BSc. And also we are trying to containerize our core tools to let the users to run our core tools in a highly reproducible way to understand, to reduce this kind of problem when you have several dependencies. In our case, we try to install our tools in different supercomputers to test what could happen. But also you can use these tools in your computer, of course, because this, as I was mentioning before, these are tools that are widely used by the community. And using these kind of containers is like very important to increase the reproducibility in science. So in this case, the workflows are not only, let's say, describing a recipe to solve a real use case, but they also provide like a second layer of parallelization, because if you want to do the same for 100 patients, it's important that the core tool could use distributed computing to use like different machines, but also from the point of view of the workflow, it's quite easy to distribute this computing, no? So if we manage to have like a given simulation or a given analysis that could use like one entire supercomputer, this will mean in the future that we will have like very complex and very, very accurate models. So this is the type of workflow that we are working in. We usually employ single cell sequencing to characterize the different cell types that we have in our simulations. Then we use this information to personalize the Boolean models or the molecular pathway that we are introducing in the simulation. And then we do the simulations. And as I was saying, you have several patients with different subtypes or with different phenotypes. You can do like a meta-analysis to understand what are the main differences. So we are more or less applying the same workflow, the same strategy to different use cases. And I can summarize some of them. For instance, we are working in simulating how a tumor is growing. This is like a very challenging approach because you have to take into account not only, of course, the tumor cells, you have to take into account also the role of the immune cells. There are immune cells that try to actively fight against the tumor, but as you may know, there are others that help the tumor, for instance, to create vascularization. And of course, there is like a kind of microenvironment that is quite complex, and we would like to simulate this to understand properly a given tumor. We had also a COVID-19 use case because of the pandemic situation. In this case, we were doing simulations to understand how the epithelial cells of the lung are being infected by the SARS-CoV-2. And the idea is to find like some targets that could stop this proliferation and could impede the SARS-CoV-2 to internalize into the epithelial cells, right? So this is more or less the same approach, but in a different use case. The third use case, we are also like working with drugs, trying to find like some drug synergies. This is something that you could do directly with the Boolean models, but if you have this kind of tissue information, you can understand how the drugs are being distributed across the extracellular space. And this is a very interesting task, and we were finding like different drug combinations that could be very useful to treat some disease. In this case, we work with cell lines, and we have a very good combination of experiments, and this is a kind of workflow also that takes advantage that is being benefit of using Carnival, for instance, to infer different molecular pathways and using MABOS then to analyze this. And we are not only, of course, to implement use cases. We are very, very interested in doing benchmarking, in understanding where are the bottlenecks and what are the differences. In this case, we are doing benchmarking from the computational level to understand what are like the resources that every single tool are consuming. Like, for instance, how much memory you need to use when you are using this tool, but also how many, I don't know, computational resources to use. In this case, it's also important to take into account the differences from the scientific point of view. Of course, in our particular case, what we try to do is to collect other tools that are similar to our core tools. We selected our core tools because they are widely used, but we know that there are many others that are very useful. And in this case, when you collect different tools for doing the same, for instance, for doing the cell level simulations, you immediately see that these tools are quite heterogeneous. It's not like they have like the same input and the same output. No, if, for instance, you are working with variant colors or with green mappers, usually you have the same input and the same output. In our case, it's quite difficult. And that's why we are doing the benchmarking not by our own. We are doing the benchmarking directly contacting the developers of these tools and trying to do like a kind of community benchmarking. And we think that this is very useful because this is a kind of activity that could help to improve a given field, that could help to define at least some standards. And of course, you can see like some limitations on some particular tools that could be improved in the future. So, and then comes PerMedCoE 2. Of course, what I talked already, it's like a bunch of different strategies and different tasks that we were doing. In this case, we want to expand this to take all the work that we were doing in the first 3 years and then expand it to improve a bit more the personalized medicine. We are focusing on different goals. For instance, one of the most important focus for us is modeling organoids. As you may know, organoids are like simplified versions of real organs. For us, they are like a kind of ideal systems to calibrate our simulations and, of course, to validate the predictions that we can do with our simulations. And, of course, from the ethical point of view, they are very important. The organoids have several limitations compared, for instance, to mouse models, because the mouse models are like whole body models, but they are usually patient-derived cells, and right now they are able like to recapitulate the complexity of different organs, like, for instance, kidney organoids or heart organoids. And we think that this is very important in order to help to reduce and to, in the future, to eliminate the animal-based studies. The second part, of course, is the use of artificial intelligence. As you may know, we are like in a new era of artificial intelligence. We have like language models. There are like many different perspectives that they are helping a lot to think that we can expand a bit more the computational biology that we are doing. In this case, we can use artificial intelligence in many different parts of our pipelines. We can use artificial intelligence to help inferring better models, better molecular pathways to then use the simulations. But we also can use this artificial intelligence to analyze the output of the simulations. For instance, you have two different subtypes of patients, and the difference, for instance, is how a given molecule is distributed across the space. So these kind of results are very complex. It is not quite easy to understand these kind of differences, and we help that with the use of artificial intelligence and the explainability of artificial intelligence. We can like model these kind of situations. And the third part is, of course, to use appropriate interfaces, so to bring our interfaces to different types of users, of different communities. Of course, the work that we are doing, we think that it will be very important for the future of personalized medicine. But if we don't manage to have user interfaces for those users that are more close to the clinical, so it's going to be useless completely. So we want to combine these all scientific communities, and we want to provide like some interfaces for those that have more advanced skills in computation, that know really well how to connect to a supercomputer and to run a workflow. But we also need to have like simple interfaces or friendly interfaces to those clinical environments, and help it to do like the same powerful simulations, but with very simplified interfaces. So these are like the main goals of our project. We call it PerMedCoE hallmarks. These are like defining the future of our project. And just to finish, you are interested in the dissemination activities that we are doing. We have like different social channels. We have information in Twitter and LinkedIn, but we also have YouTube, where we have some videos explaining how our core tools are working, or how is the work that we are doing. We have a booth also at the ISMB, so if you want to know more details, you can talk with us. We're also doing some training activities to help like the future generation of researchers to understand what are the benefits of the use of HPC and the modeling. And in this case, we are doing like some webinars that help really well the people to understand all these fields. But we are also organizing courses, for instance, to bring HPC to life scientists, right? We are also very proud of our first summer school. We received like very good evaluations from the students there. This was very funny, and we were also teaching a lot of things about our core tools, and we plan to do exactly the same for the next year. And this is very exciting for us, because this is like the first generation of PhD students that will grow professionally, understanding the relevance of having high performance computing and to having this kind of modeling for the future of personalized medicine. And you are interested in this, we are also organizing an ECCB workshop. That is a kind of very prestigious workshops. This is in November. If you want to know more information, you can follow the QR. And if you are a student, a motivated PhD candidate, of course, we are trying to find new people for developing this kind of tools. So if you are interested, please contact us. Just to finish, thank you very much. Thank you all the people. I would like to thank you all the people of the project for doing this kind of strong effort. I would like also to thank other people that are not formally belonging to the project, but are helping or are supporting the project, like for instance, those researchers that are doing the webinars, these kind of things. And of course, thanks to all of you for attending the seminar. Thank you.